Hello everyone, I am Dr. Geeta Hegde and today we will be learning about transport mechanisms across cell membrane. Transport mechanisms across cell membrane are crucial for maintaining cell function, cellular function and homeostasis. Now, at the end of this session, the student shall be able to answer about passive transport, simple and facilitated diffusion, osmosis, active transport, primary and secondary, vesicular transport. So there are three different types of cell transport: passive transport, active transport and vesicular transport. Now what is passive transport? There are characteristics of passive transport. It is the mechanism of transport of substances along the concentration gradient. It is also known as downhill transport and it depends on concentration gradient, electrical gradient and pressure gradient. Let us have a look at the types of passive transport. The types of passive transport are simple diffusion, facilitated diffusion. So what is simple diffusion? Let us have a look at it with the help of a diagram. Simple diffusion is the movement of molecules or ions from an area of higher concentration to an area of lower concentration. This continues till equilibrium is reached. And this process relies solely on the natural kinetic energy of the molecules and does not require any external energy or specialized transport proteins. Substances transported along the concentration gradient are lipid soluble substances like oxygen, carbon dioxide, nitrogen gases, fat soluble vitamins, fatty acids, steroids. Diffusion of weak acids and bases also happens by simple diffusion. The lipid bilayer is selectively permeable. It allows non-polar molecules like oxygen, carbon dioxide and small uncharged molecules to pass through more readily than polar or charged molecules. Over time, the concentration of the molecules becomes evenly distributed on both sides of the cell membrane. This is known as equilibrium. So, simple diffusion is movement of molecules or ions from an area of higher concentration to an area of lower concentration till this equilibrium is reached. The second type of passive transport is facilitated diffusion. Now, facilitated diffusion involves specific proteins in the cell membrane that aid or help in the transport of molecules or ions that cannot diffuse freely through the lipid bilayer. Now, this may be due to larger size charges present on the ions and polarity. So, facilitated diffusion happens through protein channels. Now these protein channels help in movement of larger molecules which cannot pass through by simple diffusion. Now this facilitated diffusion can happen through channel proteins or carrier proteins. Here is another example of water channel which allows movement of water molecules. These are also known as aquaporins. Facilitated diffusion can happen either through channel proteins or carrier protein. In the first diagram, we can see that facilitated diffusion is happening through channel proteins. So, what are channel proteins? These channel proteins are proteins which form hydrophilic channels that span across the membrane, allowing specific ions or molecules to pass through. What is the function? These channels can be open all the time. They are known as leaky channels, or they may be gated channels which may open or close in response to specific signals like changes in voltage or binding of molecules. For example, potassium channels allows only potassium ions to move across the membrane, which is crucial for maintaining the membrane potential. The second is facilitated diffusion through carrier protein. Now what are carrier protein? These carrier proteins bind specifically to the molecule which they transport. They undergo a conformational change. And once the carrier protein undergoes a conformational change, this helps in shuttling the molecule across the membrane. The function is, unlike the channel proteins, the carrier proteins are not always open. They operate through binding and release mechanism. Example of carrier protein type of facilitated diffusion is glucose transporter. Now what is glucose transporter? Glucose transporter, a specific type of carrier protein is GLUT, G-L-U-T, which facilitates the uptake of glucose into the cells by binding glucose on one side of the membrane, undergoing a conformational change and then releasing the glucose on the other side. Now, the channel proteins can be different types of channel proteins. In the first diagram we see there is simple diffusion happening. In the second diagram we see facilitated diffusion which is happening. In the second diagram there is facilitated diffusion happening through an ungated channel, and in the third diagram we see gated channel which is opening and closing. Third type of passive transport is osmosis. So what is osmosis? Osmosis is also a type of facilitated diffusion. It is specific to diffusion of water. Now this involves movement of water molecules through a semi-permeable membrane. So the water molecules move through the semi-permeable membrane with the help of specialized water channels which are called aquaporins, or they can directly move through the lipid bilayer. Water moves from areas of low solute concentration to areas of high solute concentration till the time equilibrium is achieved. Now what are aquaporins? Aquaporins are integral membrane proteins that form water specific channels, allowing for rapid water movement compared to simple diffusion through the lipid bilayer. Now this is about passive transport. Let us have a look about active transport. Now what are the characteristics of active transport? Mechanism of transport can happen against the concentration gradient. It is also called uphill transport. Because this is happening against the concentration gradient, it will utilize energy. So this process uses energy derived from hydrolysis of ATP molecule to move ions or molecules against their concentration gradient. It directly involves ATP driven pumps. So this process also shows saturation. It is specific for particular molecules and ions. So these are the characteristics of active transport. Active transport is a mechanism of transport against the concentration gradient. Hence it is also called uphill transport. This process uses energy derived from hydrolysis of ATP molecule to move ions or molecules against the concentration gradient. It is also known as ATP driven pump. So ATP is broken down into ADP and an inorganic phosphate, releasing energy. This energy is used to change the conformation of the transport protein. Okay. So active transport is uphill transport. This process shows saturation. It requires energy and it is specific for molecules and ions. It is the mechanism which is happening against the concentration gradient. So it also shows competitive inhibition. Now one type of active transport is primary active transport. Now in this primary active transport we see that there is a carrier protein which will undergo a conformational change, and this conformational change will only happen after ATP is broken down into ADP, which is known as ATP hydrolysis. So ATP hydrolysis releases the energy. This energy is used to change the conformation of the transport protein. This is known as primary active transport. Another diagram to show about the primary active transport. Primary active transport is the most common pump found in all parts of the body. It is an electrogenic pump. It is responsible for maintenance of resting membrane potential of all the cells, and it also helps in maintaining the cell volume. So the channel which we see here, the primary active transporter which we see here, is also known as sodium potassium pump. So this sodium potassium pump pumps 3 sodium ions out of the cell and 2 potassium ions into the cell against their respective gradients. So it is against the concentration gradient, and this sodium potassium ATPase pump is crucial for maintaining the electrochemical gradient necessary for various cell processes. Now where is it needed? It is needed for nerve impulse transmission. It is required for muscle contraction, and it is also required for maintaining the cell volume. This is the same pump which is responsible for maintaining the resting membrane potential. So I'll repeat again. Primary active transport is a type of active transport. This process uses energy derived from hydrolysis of the ATP to move ions or molecules against the concentration gradient. The most important example of primary active transport is sodium potassium ATPase pump. It is also known as sodium potassium pump. What does this pump do? This pump pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. Hence, since it is moving 3 positive and outside and taking only 2 positives inside, this is an electrogenic pump. This pump is crucial for maintaining the electrochemical gradient which is necessary for nerve impulse transmission, muscle contraction, maintenance of cell volume. It is also responsible for the resting membrane potential. Another type of active transport is secondary active transport. So what is secondary active transport? This is the type of transport which uses the energy stored in the form of an ion gradient. Now how is the ion gradient established? It is established by the primary active transport to move any other substance across the gradient. Now there are two types of secondary active transporters. One can be symporter, other is antiporter. So what is symporter? Symporter, as we can see in the first diagram, it is sodium potassium chloride co-transporter or symporter. In the second example, it is potassium chloride co-transporter. So potassium chloride co-transporter is transporting potassium and chloride ions both in the same direction. In the first is sodium, potassium and chloride, all three are transported in the same direction. In the third diagram, which is another type of symporter, sodium, GABA and dopamine, that means sodium is helps in transportation, co-transportation of the neurotransmitter dopamine and GABA. So symporters transport both the ion as well as the another molecule in the same direction across the membrane. What do the antiporters do? Antiporters move the driving ion and another molecule in opposite diagram, in the opposite direction. For example, sodium calcium exchanger, sodium is moving inside while calcium is moving outside. So it is both the ions are moving in the opposite direction. Hence it is antiporter. Another type of antiporter is sodium hydrogen exchanger. Here also sodium ion is moving inside and hydrogen ion or proton pump is moving outside. The third type of active transport is bulk transport. It is also known as vesicular transport. So what is vesicular transport? This involves the movement of large particles, liquids or macromolecules in vesicles. Now what is a vesicle? Vesicle is a membrane bound sac. Now if this membrane bound sac helps in moving the substance inside, it will be known as endocytosis. If it is moving the substance outside, it will be known as exocytosis. Okay, so endocytosis again is of three types: phagocytosis, pinocytosis and receptor mediated endocytosis. I'll repeat again. Vesicular transport or bulk transport involves movement of large particles or liquids or macromolecules in vesicles. What are vesicles? Vesicles are membrane bound sacs. This process requires energy, that is why it is a type of active transport. And why is energy required? ATP is required for rearrangement of the cell membrane. Now the first type of vesicular transport is endocytosis. Endocytosis is of three types: phagocytosis, which is cell eating; pinocytosis, which is cell drinking; and third is receptor mediated endocytosis. So in the first example we see about phagocytosis. So what is phagocytosis? The cell membrane, as you can see in the diagram, the cell membrane engulfs a large particle or cell forming a phagosome. It forms pseudopodia around the particle which has to be engulfed inside. This is known as pseudopodia and it leads to engulfing of large particle or cell forming a phagosome. As you can see, phagosome is also known as a food vacuole. So what does this phagosome do? This phagosome then fuses with the lysosome to digest the engulfed material. So where is this phagocytosis used? Phagocytosis is an example of active transport, endocytosis, which is used by phagocytic cells. Now immune cells like macrophages and neutrophils engulf pathogens or debris. Second type of vesicular transport is pinocytosis. Pinocytosis is also known as cell drinking. So the process is the cell membrane invaginates, as you can see in the first step, the cell membrane invaginates to form a small vesicle. This vesicle contains extracellular fluid and dissolved substances. Then these vesicles are internalized. For example, cells absorb extracellular fluid and nutrients. Okay, so this is known as cell drinking. Phagocytosis is cell eating. This is cell drinking. Phagocytosis, pinocytosis are both examples of endocytosis. The third type of endocytosis is receptor mediated endocytosis. Now what is receptor mediated endocytosis? Here specific molecules bind to the receptors. As you can see the receptors, the specific molecules bind to those receptors on the cell surface. Now once they bind to the cell surface, this triggers membrane invagination and vesicle formation. Then the vesicle is internalized. So this step is similar to phagocytosis, but it requires presence of the receptors, and once the specific molecule binds to this receptor, then it triggers membrane invagination and the vesicle is utilized. The example is uptake of cholesterol via LDL receptors. Okay, so this is the third type of endocytosis. So where are we? We were talking about passive transport, active transport, and third is vesicular transport. Vesicular transport is also a type of active transport. There are three types of vesicular transport. One is phagocytosis, which is cell eating. Second is pinocytosis, which is cell drinking. And third is receptor mediated endocytosis. The example for receptor mediated endocytosis is uptake of cholesterol via the LDL receptors. The fourth is exocytosis. Now exocytosis is a type of vesicular transport, so this also requires energy. Now what is exocytosis? In exocytosis, the vesicles containing substances fuse with the plasma membrane. There are newly formed vesicles which contain the waste matter inside the cell. Now this waste matter has to be thrown out, so the vesicle containing substances fuse with the plasma membrane, and this releases the content into the extracellular space. This process is crucial for secretion and membrane recycling. Also, this exocytosis is also used for release of neurotransmitter from nerve cells, secretion of hormones from the endocrine cells, and export of cellular waste. Thus, all these cell transport mechanisms ensure that the cell can efficiently exchange substances with their environment, maintain internal conditions, and carry out necessary physiological function. So, towards the end, let us learn about the differences between passive and active transport. Passive transport is along the concentration gradient. It is known as downhill transport. Here energy is not required. Process continues till the time equilibrium is achieved. There is no specificity for particular molecules and there is no competitive inhibition. As opposed to active transport is always against the concentration gradient. Here energy or ATP is required. ATP undergoes hydrolysis, and this energy which is released from hydrolysis of ATP is used to produce the conformational change in the carrier protein. This is also known as uphill transport, and the process shows saturation. There is specificity for particular molecules and ions, and this also shows competitive inhibition. So to summarize, today we learned about the various cell transport mechanisms. The different types of cell transport mechanisms are passive transport, active transport, and vesicular transport. Passive transport is again of two types: simple diffusion, facilitated diffusion. This also involves osmosis. So passive transport includes simple diffusion, facilitated diffusion, osmosis. Active transport is of two types: primary active transport and secondary active transport. Secondary active transport can again involve symporters or antiporters. And the third is vesicular transport. Vesicular transport is of two types: endocytosis, exocytosis. Endocytosis is again of three types: phagocytosis, which is cell eating; pinocytosis, which is cell drinking; and third is receptor mediated endocytosis; and last is exocytosis. So vesicular transport is of two types: endocytosis and exocytosis. I hope this lesson was useful for you all. Thank you.